So what we know is that there is an increased selective attention in spider phobics viewing spider pictures, and it starts already at 200 milliseconds after the onset of the stimuli. We also know that this increased attention to spider pictures and spider phobics is similar to other emotional pictures, so it's not fear specific. You can see this increased activation in the visual cortex, in spider phobics is probably, wait a minute, sorry, okay. This is amygdala. A colleague of mine, Julia Wendt, presented pictures to spider phobics in the scanner, and she observed that the amygdala was more activated during spider pictures viewing in spider phobics when compared to mushrooms. This is here and here, and there were no difference in controls. So we know what we know from the scanner studies, that there is a amygdala activation and visual cortex activation, and this is probably, it looks like, I didn't have this, wait a minute, okay, it is probably like you have the activation in visual cortex, and it is driven by amygdala, because we know from animal studies that the amygdala activation occurs earlier, and this is probably the cause of increased tension and increased activation in the visual cortex. So, what about psychotherapy? How can we use this knowledge actually? It's far away, it seems to be. So, as I told, increased selective attention is not fear specific, but it's emotionally specific. And there are some ideas to change attention when working with anxiety patients. So you can imagine, you are scared of spiders, so don't look at spiders. Think about something different. It's better because it's already emotional regulation at work sometimes, but not in case of phobia. It's not in the case of specific phobia. So the point is not to change attention during the therapy, but to change emotion. We need to change fear directly, not through changing attention. How to do it, I will tell you. I hope I won't forget. The next point is, during the therapy of anxiety disordered patients, it is very important to look at the risk, because they are very sensitive, to look at the risk of the fear system sensitization and the fear system conditioning during the therapy. I'll show you a bit more now. Why? This is the first study. I showed you the EPN and LPP, and now you can see a component earlier time. Here you have spiders, and as you can see, also like earlier, thick line, these are earpiece for spider phobics, and thin earpiece in control group, and you can see increased P1 and spider phobics looking at spiders, but this is an unspecific reaction. You can see an increased P1 in each picture, during each picture presentation. We think it's something like hypervigilance, and we also think we are now investigating it in scanner, because we don't know whether it comes from the amygdala, but we have a hypothesis that this increased vigilance, because it is actually increased vigilance. They are already vigilant because they are expecting something anxious. That this increased vigilance is also activated by the amygdala. But we don't know whether, actually in this experiment, we cannot say whether this was caused by the expectation of spiders, or this is just a feature of spider phobics, or they expect something threatening, not spiders, but generally something threatening, because in this presentation they saw different pictures. So we don't know what's the cause of the P1. We have the hypothesis, this is the P1, the vigilance caused by the expectation of spiders, but we don't know, actually. So, we don't know. Is it specific to the phobia-relevant threat context, the increased hypervigilance, or it's specific to general threat context, they are just more vigilant and more sensitive to different threat context, or it's unspecific. Maybe they are looking at neutral pictures and they already have greater vigilance, we don't know. So we made an experiment, a next experiment for the same group, but we showed them two different blocks. One block was a neutral block with only neutral pictures, and the second block, always the second block, was the block with spider pictures. We wanted to look whether they will show an increased P1 already here during neutral picture presentation, or not. It would speak when they wouldn't show this increased P1 in the neutral picture presentation. It would speak, okay, this first result was the reason of expectation of spider. But the problem was here. We didn't publish it because it was a little bit problematic. The problem's here. Here, this the safe context where we saw when they saw only neutral pictures, and here this is the spider-relevant context when they saw spider pictures. And as you can see here, in the P1, no difference. But we didn't believe it. We tried once more. It would speak for a general hypervigilance in spider phobia. The theories say something different. So we tried once more, same group, but we created a cover story. Because we thought they already knew, because they were screened with spider phobia questionnaire. They already knew they will see some spiders here. They were just normal students. Students can expect something like this. So we came to the lectures, different lectures. A group of people came to different lectures at the University of Warsaw, and they made a screening with spider phobia questionnaire. And then, 4 weeks later, I came to the same lecture, and I made a screening with some test for attention styles, I think it was. And then I chose people on the basis of the SPQ, spider phobia questionnaire. But I called them and told them, okay, you have this, I've been there to your lecture, and you have this attention style, we need you. And I invited them for participation in the study. And what we showed was a little bit different as in the study before. We showed them a neutral block. We showed them a block with threatening but not spider-relevant pictures, and we showed them a block with spider pictures. So we had 3 conditions, we counterbalanced them, and let's see, it's complicated here. Okay, I will try to do it. So, look here in the middle. This is the replication. This is similar to our expectations. The red lines is the P1 in spider phobics, and the black ones, this is the P1 and controls. So you can see here in the spider, during the spider exposition, the spider phobics show increased P1 and compared to controls. So it is in line with our expectation. They may be afraid of spiders, so they show increased hypervigilance. But interesting is, and if you look here, that in a safe condition, when we only show them neutral pictures, there were no difference between red spider phobics and black controls. And even in the non-phobic threat condition, this was the block when we showed them neutral and unpleasant pictures, but not spiders. There was no difference. But only if, for people who saw these presentations before the exposure to spider pictures. As far as they were told they will see some spiders in this experiment, so after this, after we uncovered the story, they also showed increased P1 in both safe and non-phobic threat conditions. So, even though we told them there won't be any spiders in these conditions, so actually they were already sensitized during the spider presentation, and it is a long-lasting sensitization. So they saw this hypervigilance also later on. Okay. So we already know that spider phobics show selective attention towards spiders, and we know that they have an increased hypervigilance when they are exposed to the phobic threat context, and that this increased hypervigilance, this is an effect that takes longer. Even though they are told they are safe, they don't feel safe. They show hypervigilance. Okay, now let's look what can we do about it. There are data that these are animal data, that this long-lasting sensitization of the fear circuit. We still don't know whether this is a fear circuit sensitization, because we only can see the surface in the EEG, but we will investigate. So there are data that the sensitization of the fear circuit may result in an increased fear conditioning after. So, I mean, when they are exposed to some threatening contexts, these were rats' data. When rats are exposed to food shocks, they can be easier conditions in another context afterwards, and they show more avoidance behaviors in other contexts. So, okay, let's come to the conclusion. So, these mechanisms, if we do something like this in the therapy, I mean, during the therapy, we need to talk about things they are scared, yeah. But there is a risk that if we talk about it and if we show some things to some events or situations they are scared of to our patients, then we get this effect, and they may, imagine you go, you saw some horror film, and then you've been to a friend, and then you go out. And the situation is different when you go out after a horror film and after a comedy. Yeah? And you need to walk through a forest, so you are more hypervigilant. And if we get our patients to the state that they are more hypervigilant, they may even get a new fear response to different stimuli. And the most important thing is probably